All right, party people, we are back at it with everyone's favorite, biochemistry. Let's get it. So if I was to give you a 24-month-old infant with lethargy, she's unresponsive with tachycardia, hepatomegaly, and in her blood, she has decreased glucose as well as decreased ketones. In her urine, she has decreased ketones, but she has increased dicarboxylic acids. For C6, there's mild elevation. For C8, there's extreme elevation. And C10 also has mild elevation. And I ask you, what enzyme is deficient? So in order to solve this riddle, let's run through it. We've got an infant with congenital enzyme deficiency. Lab findings show hypoketotic hypoglycemia. That means low serum ketones and low serum glucose. This should ring bells for some students. We also have decreased urinary ketones and increased dicarboxylic acids. Now, you might be wondering what is meant by dicarboxylic acid. These are just molecules with two carboxylic acid functional groups. The chemistry itself isn't that important here. It's just something you will see in this particular condition. Then we have to address the acylcarnitine profile that was given. The key point here is that it shows a spike in C8 carnitine, that's octanoyl carnitine, along with other increases in hexanoyl carnitine as well as decanoyl carnitines. These are medium chain fatty acyl carnitine. These accumulate in MCAD deficiency. MCAD deficiency stands for medium chain acyl-CoA dehydrogenase deficiency. Why we have hypoketotic hypoglycemia needs to be addressed. So when we break down fatty acids, the process starts with the fatty acid in the cytosol approaching the mitochondria. But it simply cannot cross the mitochondrial membranes on its own. It must be first converted by an enzyme into fatty acyl-CoA. Fatty acyl-CoA is then shuttled into the mitochondrion via the carnitine shuttle. Now, once inside the mitochondrion, beta oxidation begins to break down the fatty acyl-CoA. So in MCAD deficiency, we can't properly break down medium chain fatty acyl-CoAs. And this is done normally via beta oxidation. Here it's deficient. So therefore, as a result, these fatty acylcarnitines accumulate, which is why we see elevations in C6 through C10. During beta oxidation, we normally chop off 2 carbon units, aka acyl-CoA, from fatty acids. So for example, breaking down an 8-carbon fatty chain yields 4 acetyl-CoA molecules. These acetyl-CoAs then fuel ketogenesis, producing ketone bodies like acetoacetate, beta-hydroxybutyrate, and acetone. Acetone is the one responsible for the fruity breath in ketoacidosis. But in MCAD deficiency, since beta oxidation is impaired, we don't generate sufficient acetyl-CoAs, and therefore we cannot make ketones. Therefore, you guessed it, leads to hypoketosis. Now, why is glucose low? This also needs to be addressed. Acetyl-CoA is a positive allosteric regulator of pyruvate carboxylase. This is a key gluconeogenic enzyme. Here's the sequence at the end of glycolysis. Pyruvate can either enter the Krebs cycle via pyruvate dehydrogenase, or it can go backwards towards glucose through gluconeogenesis. However, it can't just reverse directly. Glycolysis includes irreversible steps. So one workaround is that pyruvate carboxylase, which is activated by acetyl-CoA, converts pyruvate to oxaloacetate, which can then be converted into phosphoenol pyruvate, PEP, and ultimately back up the chain to glucose. So in MCAD deficiency, since we're not producing acetyl-CoA, pyruvate carboxylase isn't activated and gluconeogenesis is impaired. This results in hypoglycemia. So in summary, hypoketotic hypoglycemia with the accumulation of medium chain fatty acylcarnitines, those being the C6 through C10s. The mechanism is impaired beta oxidation. This results in decreased acetyl-CoA, decreased ketogenesis, and decreased gluconeogenesis. As far as the differential diagnosis as well as other enzymes, LCAD deficiency would present with elevated long-chain fatty acylcarnitines. Those would be C12 or longer. Carnitine deficiency means carnitine shuttle can't operate, so you'd see a buildup of fatty acyl-CoAs, but no acylcarnitines. So let's touch on some other related enzymes that show up on your licensing exams. Acetyl-CoA carboxylase is used in fatty acid synthesis, not the breakdown. This converts acetyl-CoA to malonyl-CoA. Malonyl-CoA inhibits carnitine shuttle, preventing fatty acid entry into the mitochondria. This makes sense if we're synthesizing fats. We don't want to break them down. Please note that biotin, aka vitamin B7, is a dependent vitamin for acetyl-CoA carboxylase. Next up, propionyl-CoA carboxylase. This also needs vitamin B7, aka biotin. It's involved in the breakdown of odd chain fatty acids, and it converts propionyl-CoA to methylmalonyl-CoA, which then becomes succinyl-CoA, which is B12 dependent. It then results in the entry to the TCA cycle. As far as hormone-sensitive lipase versus lipoprotein lipase, for hormone-sensitive lipase, this is catabolic. It mobilizes triglycerides from adipocytes and moves them into the blood. This is stimulated by cortisol, glucagon, and epinephrine. As far as lipoprotein lipase, this is anabolic. It brings triglycerides from the blood into adipocytes, which is stimulated by insulin. It's also upregulated by fibrates, such as gemfibrozil or fenofibrate. This is the first-line treatment for hypertriglyceridemia, which is over 500 milligrams per deciliter of triglycerides. As far as type 1 familial dyslipidemia, this is due to deficiency of LPL or ApoC2. This leads to elevated chylomicrons. All right, that's it for this one. Go crush it.